continue using it after the course, the demo server, even if you need to re-ask for a new account because they're going to do some sort of migration or something. But still, it's really worth it, I think. Okay, so I will talk now about publishing images. So you see we're getting towards the end of the course quite clearly because we are talking about publications now. So that's another topic. So one thing that I would like to say is when you want to publish something, you should start by reading the journal guidelines about your images. Oh, I realize I'm not finished with my coffee. Some things have to come first. Okay. So yes, read the guidelines about images. It's very important because different journals have different guidelines. So don't make your figures and then look. So there's a nice online tool here to how to deal with images. There's also the JCB, the Journal of Cell Biology guidelines, which are quite detailed. And there's quite a lot from JCB. They used to have a blog. So there are 10 very nice blog posts from JCB that you should read before you make a figure. So I have all these links in here. And if people in abroad, whatever, people online are interested, I can put them in the chat at the end. Just let me know. So last year, there was a big step, let's say, into trying to improve standards for light microscopy. You may not be aware of this initiative, but it's a very big international initiative called QUAREP-LIMI. And that stands for Quality and Reproducibility in Light Microscopy. And the QUAREP-LIMI had different work groups that work on different things. And I participated in, a few of them, but one of them published last year. And so the paper is called Community Developed Checklist for Publishing Images and Image Analysis. It's in Nature Communication, and I have the link in, or I have actually uploaded the PDF in Canvas in the page for this lecture. So you're welcome to download it and have a look. And the way it works, these are guidelines from when you acquire images and then you have the images, the microscopy images, and you want to get the result and publish them. So it's this workflow in here where the paper is trying to set some standards, let's say. And the idea is that we have 3 different levels: a minimal requirement, recommended requirement, and ideal requirement for each for each type of feature, let's say, that you need to check. And so this is presented like this as checklists, where you can yourself, when you want to publish a paper, you can look at the checklist and go, so like, okay, am I showing example used for the quantification? Yes, etc. And so the minimal is shown like this. And otherwise, some things are recommended. So things about, putting the images in black and white. It's one of the things that I've been saying here. Many other stuff as well. So, getting a scale bar and what is the resizing, rotating, and all this type of things. Quite a few of those I'm going to talk about now. But in these checklists, I really recommend that you have a look. And you have a look exactly before you start making your images, so that to help yourself. Okay, so when we want to acquire an image for publication, there are many different things that we have to check. So one of them is the intensity. And as you know, we should have no bleed through, no saturation, no underexposure in the area of interest. This is very important. We need to make sure that we fill the histogram as much as possible. So the maximum intensity in the image should be, if possible, close to saturation, but under. So we should have used the dynamic range of the image as much as possible, or the bit depth, I should say, as much as possible. And then the minimum pixel, so the dimmest pixel in the image should be just above the 0. So this way we really use the full bit depth of the image. And again, you can have a look at the histogram in Fiji by clicking on Control H, and you get this histogram, and that tells you in here the bit depth at the top of the image. So 8 bits means from a 256 level of gray, so from 0 to 255. And in here in Fiji, in the histogram, you can see the minimum and the maximum in the image. And so this image looks very dim, but it is actually correctly exposed for an 8-bit image, because we have the minimum just above 0 and the maximum just below 255. So we can then, so means this image is acquired in a proper way, and then we can use the brightness and contrast to display, and the gamma to display this image differently. The images that you use to compare intensities must be acquired in the same way. And it doesn't mean they have to be displayed in the same way, but they must be acquired in the same way. And then you can adjust the acquisition settings for the brightest sample. So if you have bright and dim samples, use the brightest sample to adjust the acquisition settings so that you avoid saturation. So this is really important. Saturation and underexposure are artifacts of microscopy. Your data are not correct, so please don't. Okay, another aspect of publishing an image is the orientation. So if you rotate an image, so it's possible in PowerPoint, for example, to rotate an image, but that leads to extrapolation and therefore distortion of the image. And you might not see it, but the image might, for example, not look that good when it's published. And also it might be visible if you have not so many pixels in your image. Only a rotation of a multiple of 90 degrees, this is, the pixels are squares, right? So if we rotate it by any angle except a multiple of 90 degrees, we do distort the image. So if you rotate by 90 degrees, then no distortion. We have at our facility some homemade, not homemade, but custom-made stage inserts that allow us to rotate as we want the sample itself during acquisition, so that we make sure that it is, for example, horizontal or vertical, and that helps a lot. So orientation, please don't rotate except if it's more than 90 degrees. So you have to think about it before you acquire the image, okay? Another aspect is the image resolution. And as we know, the image resolution is either the optical resolution delivered by the microscope or the digital resolution, whichever is limiting. So we use different resolutions for different purposes. When we acquire an image at the resolution needed to, so we need to acquire the image at the resolution that is needed to extract reliable data, not beautiful images. So please just get away from trying to acquire the best images, the most beautiful. What we want is extract reliable data. So you need, whenever you push the capture button, you need to think of what you want to show in this image. And then if you want to capture an image specifically to put an image in the article, then you have to think about what you want to show. So clearly, if you want to put an image, it's because you want to show something, the image needs to show it. Okay, you will be surprised the number of articles where you have an image that is almost completely black, for example. And the person says, here you can see this and that. So so we need to show what we want to show, basically. If the image you analyze are the type of images that are, for example, very grainy and it's difficult to see things, it's totally valid to extract data from grainy images. So acquiring images that are grainy, as long as you get reliable extraction, is not a problem at all. But maybe this is not the image that you want to publish. So if you want to show an example of the images that you're analyzing, it's totally okay to publish an image that is grainy. If, however, you want to say, well, in these images, this is a representative image, but the grain, the noise is basically hiding what you want to show to the person who reads the article, then it's totally okay to acquire the images in a different way. But as long as you explain it in the material method, it's not an issue. So in the case when you analyze the images you analyze contain reliable information, but they do not show clearly what you want to convey, also acquire images that show what you want the viewer to see. And you can imagine that you have to think about it when you're at the microscope and you're imaging. So just try to also acquire an image that is basically showing what you want to show. In that case, just go for it, oversample to allow magnifying in the image. Let's say, for example, you will be able to apply for having a cover image if you have an image that is oversampled. And then, so magnifying without showing the pixels. So try to use, to acquire such an image, then you need to have more pixels than 1024 by 1024. If it's possible. And then you can use high illumination, long exposure time, averaging to improve the signal to noise ratio. So basically, you know, think about what it is that I want to show when I'm acquiring this particular image, and you set all your parameters for this particular image, right? So we don't have such a thing as settings that work for everything. So another aspect of publishing images is the field of view. Clearly, this is suboptimal. We have a very small object in the middle of a huge black area. So what can we do? When can we crop or not crop? In this image, we have these two little things that are there and that are, you know, whatever this is, background, right? Something that is there is really fluorescent and we don't want. So you need to interpret what that is. But if this is irrelevant for the interpretation of this image, then it's totally okay to remove it, to crop it, basically, so that you only show this. So when imaging on a confocal, you can increase the zoom to fill the field of view with your sample. And on a camera-based system, you can acquire the image and then crop either at acquisition time or afterwards, so that you only acquire the pixels with the relevant data. So in this image, most pixels contain no relevant information. And in here, we have the same image, but it's just cropped. So it's much easier for the viewer to actually see what you want to show. I don't know what it would be, but maybe this little dot here or the red dots in the middle. And yeah, so basically, when you decide to crop or not to crop, you just need to think, is my action now going to change the interpretation of the image or not? If it changes the interpretation of the image, don't do it, okay? If it doesn't, so if I zoom in here or I don't zoom in here, and whether I see these two little things or not, it's likely not to change the interpretation of this area, so it's okay to do it, okay? Irrelevant information is removed in this image, but there's no change in the image interpretation if it's cropped, so then that's okay. Another aspect of publishing images are the channels. I think I have said during this course, please display the images in black and white, not in colors. And if you have an overlay, you can put it in color. But another aspect is do not acquire images that are not needed. So some people on the point confocal, when you image a transmitted light, all you need to do is turn on the transmitted light detector. And so that means that you have that you're going to have an extra channel that maybe you don't care about. So it's very easy to turn on the transmitted light detector. You don't need to have more laser or whatever, because it's whichever laser you're using for fluorescence that go through the sample, that's what you're detecting. But at the end of the day, it increases your the file size. And then it means that when you want to publish, you have to take care of removing it, for example. So just only acquire the images that you need. One typical example as well, on top of the transmitted light, is the nucleus. Ask yourself why you're imaging the nucleus. And if you're not going to use the image of the nucleus in your image analysis, don't image the nucleus, okay? So only image the channels that are needed. And clearly, no, in this particular case, the bright field is also saturated, so we can't see anything in that. The formats, the image formats. So as you know now, the images do not only contain pixel data, which is X, Y, and intensity, but they also contain metadata, And that contains a lot of information on the acquisition settings, which filters, at which Z level we were, the date and the time, and all sorts of different things about the wavelength and the gains, and everything is saved in the metadata. So the metadata is super important in the image. So the channel color, the pixel size, the illumination power, etc., etc. And this is the reason why when you go to your microscope, very often you have a magic button. You open the old image that you did like 1 month before, and then that you acquired 1 month before, and you have a button that you can click and it resets all the settings in the same way. And this is because the software can read the metadata. So, so please do not throw away the metadata. It's very important. Always save in the original format that is recommended by the microscopy manufacturer. And also another thing is that there are different formats that have different levels of compression and quality. If you save in the original format, it is very unlikely that the manufacturer is going to recommend that you compress the image and lose quality, because they want you to gain the best quality out of their images. So save in the format that is recommended, and do not save, absolutely not in JPEG and not in TIFF either, except if this is the format that is recommended. And we will see why in a second. So all the intensity details, the resolution, and the metadata may be lost if you change format. So always save the images in the original format, and these are the formats for Zeiss. This is a bit of an old format. The new format is CZI. And then ND2 for Nikon, these formats for Leica, and these formats are for Olympus. And clearly there are other manufacturers as well. Do not put your images through several formats. I take my image, I make an 8-bit out of it, I put it in a Word file, and then, oh, I would like to present it to my lab, so I'd like to put it in PowerPoint, so I don't remember where the original image is, so I go to my Word file and I copy it from there and I put it in PowerPoint. Not a good idea. Every time you change format, you lose something. Go back to the original. So the images that are used for presentation on the computer screen must be 8 bits per channel. So if you have 3 channels, you'll have 24 bits, but it's 3 times 8 basically. And a computer screen can only read 8 bits, so 256 levels of intensity. And JPEG and PNG can be lossy or lossless. It depends on what type. So you have to realize that JPEG, TIFF, PNG, and etc. These are families of formats. They're not single formats. So it's a bit of a jungle. Depending on the software that you're using, you're going to get files that have a different format, and that's not very good. And TIFF files can be very large. And if you put TIFF files in a PowerPoint presentation, it can slow down the presentation. So for PowerPoint presentation in general, the way we display the image is anyway low resolution. So it's in general fine to put JPEG or PNG in a PowerPoint presentation. It doesn't slow down your presentation, but only if it's a presentation on a computer screen. So do not use these formats for posters or articles, publications. And do not analyze or make any decisions based on a JPEG or a PNG. You use, for the analysis or the decisions that you're making, you use the original format. So this was images that are used for presentation on a computer screen. But if you want to use to do an analysis of your images, or if you want to publish a poster or in an article in a journal, then you need to be aware that TIFF is not a single format, but a family of format. So you basically, when you save as TIFF, you don't really know what you're getting. So if you can avoid, do. These images can be either compressed or not. It can be lossy, which means that you lose data, or lossless. It depends on the software, basically. The metadata might be saved, but scrambled. It goes to different places, or it might be partly lost. So only save in TIFF if this is the recommended format by the manufacturer. So let's say in the order of quality, you would say number 1 is the one that the manufacturer recommends. Number 2, you have TIFF, because some TIFF formats do not lose any data. and do not compress the data. And then number 3, you have JPEG and PNG, which definitely compress and lose data. And then if you want to do an analysis, you always use the original format that is recommended by the manufacturer. For posters and publications, you can use uncompressed TIFF or TIFF with lossless compression. And then the the 8-bit, so you have to use either 8-bit or 16-bit per channel. So now if I look at the metadata, I remember that you can look at the metadata in Fiji. If you open the image in Fiji and then you press Control-I, you can see the metadata that is stored in the image. And you can see here that we have, this is an image in CZI, if I remember, yes, in CZI. So it's acquired on the Zeiss microscope, and you can see that we have tons and tons of metadata. Everything is stored in the image. There should be more than 20 lines of text, otherwise the metadata is likely lost. So the file size is also one aspect of publishing images, and you can see in here I've saved my images in TIFF, either in a higher bit depth than 8 or 8 bit, or I've saved in JPEG where you can only save as 8. And you can see here the size of the data is tremendously decreased when I save in JPEG. So basically JPEG is really, really for the web and for screen. This is what it's designed for. So we have a very high level of compression and also a low resolution. And there's a lot of artifacts as well in JPEG images. So fine for computer screen and only for that. So we have spoken already about the displaying images, and this clearly is an important part of publishing images. We acquire the images without saturation, without underexposure, but if the images anyway look quite dim, it's going to look dim in the paper. So we're going to push the brightness and contrast and possibly the gamma. So we adjust the brightness and contrast to clearly show what we want to show. So the viewer should not be struggling to try to understand the image. And do not push the brightness and contrast in such a way that it looks like you have under and overexposure. Maybe you will not have it, but if you push too much, some areas will look like they are saturated, even if they are not. And instead, I've told you that you should use the gamma. So you find in your software where the gamma setting is. And when you change the gamma, you can display bright, you can change the gamma to be able to display bright and dim areas in a sample. And this is not a linear transformation. It affects the pixels differently across the image. It is very important that you mention that you have used it in the material method or likely in the figure legend, and also you justify why. So in order to show the dim and the bright area, we have set the gamma to whatever, 0.4, something like this. And it takes very little, and it allows the viewer to actually see what you want to show. Colors, I've said before, our eyes are really color biased. So on top of that, clearly there are some people who have a problem with colors. It's not rare at all. So please display your images in black and white so that people can judge them without color bias, and then you put the overlay in color. And yes, if you clearly identify which elements you are referring to, for example, in the paper text or in the figure legend. So put some arrows, things that are very clear in your images. And computer screens have very different settings from one computer to another. So beware of trying to make the image look nice on your screen, and then when it goes to somebody else's screen, it looks horrible. So do not push things to their maximum. It could be that your screen is too old and it's too dim or something like this, and then you push it on yours and then it looks horrible on anybody else. So just be gentle with the way you display your images. Color blindness is 1 in 12 men. So this is 1/6 of the whole population. It's a lot of people. And there are many different types of color blindness. It's not always green, red. So they are online. You can find instructions on which colors to use. Green and magenta instead of green and red can help. Clearly, put your images in black and white anyway, but for the overlay, you can change the colors so that it works for colorblind people. And you will see when you want to publish your images that different journals have different requirements for the color space, what it's called. So we have basically two main color spaces. One is called RGB for red, green, blue, and the other one is CMYK, which stands for cyan, magenta, yellow, and black. And so CMYK is mostly used for printing on paper, and the RGB is mostly used for computers. So you don't want to be doing all your figures in one color space and then figure out that your journal wants something else. So have a look always at the requirements for the journal you're targeting. And CMYK is not very good at reproducing fluorescence, but it's much better for printing. So see what is required. Depending if your journal is purely online, it's likely that they are going to require RGB. And if they are online plus paper, then likely they will ask for CMYK. So only convert to CMYK after you've made the figure. So you do all your figure in RGB so that you can judge on the screen. And then afterwards, when it's finished, you put it in CMYK. Okay, so it's basically this presentation. I'm going through all sorts of little small aspects of publishing an image. So now, how do we represent the volume in an article, which is flat? So we have different ways to represent volume. For example, one of them is the orthogonal view, and this is available on all software and also in Fiji. And this is a very good way to show, for example, if you have a vesicle inside the cell. So you want to show that the vesicle is really inside the cell and not sort of stuck on the membrane on the outside. And so you take like a very nice Nyquist Z stack with a high numerical aperture objective of the cell, and you display the image this way. And in the center here, we have an XY image. So this is basically one of the Z levels. And then on the side, we have an XZ and an and a YZ projection. So you can see here the volume as seen from the side, basically. And so that means that you can here show, so if you can find a Z plane here, a Z plane here, and an XY position here where you can pinpoint to the vesicle that you want to show it's inside the cells. If in the 3 dimension of space it's inside, it shows that it's inside, then it means it's definitely inside. Okay, so that's quite a nice way to display something when you want to show that an object is inside another. Another way to represent a volume is to do a tile view of the Z stack. Sometimes it's useful. And we can also do, for example, a maximum intensity projection. There are other types of projections. This is one of them, and it's very commonly used. So in a maximum intensity projection, we have a Z stack. And so for each pixel in this image, we have several images, right? So we have a column of pixels because we have a whole volume. So for each pixel, we have several pixels, basically, one for each Z level. And what the software does is that it finds which is the brightest, and it keeps this for the maximum intensity projection. So for each pixel, we're going to get the brightest pixel in the column of pixels. And so that sort of flattens the image. It makes an image that has only one dimension, but where in each pixel we have the brightest in the whole volume. It means that if you have 2 vesicles or something like this that are exactly on top of each other, clearly they will become one, because only one pixel can be represented. But if the two vesicles are like this, it may become like one bigger vesicle. So there are some disadvantages in using a maximum intensity projection, but it also has the advantage that we can represent something that is a volume flat, as long as we don't have too much of that problem that things overlap. If this is the case, we cannot use maximum intensity projection, but otherwise it's a very good way to represent the volume, and it's very commonly used. So all software have maximum intensity projection. There's also in Fiji. And clearly, when you display a maximum intensity projection, you must mention it in the figure legend. It's often called MIP. So for example, if your sample is not completely flat and you're actually acquiring a big volume, this is a way to sort of make it flat and display it in one layer. Representing time. So now let's say you're doing a time lapse and you would like to represent this. Clearly, one way is to have a video in the supplementary material, but sometimes you would like to get the information, if it's like very important information, you'd like to get it in the main article as well. And so it's the same thing as for Z. We can represent time with tiles the way we have in here. So that's one possibility to show the main events, let's say, in the time lapse. Another way is to use a kymograph. And this is a kymograph. So basically all the tiles are, well, sorry, this is a kymograph and these are the tiles corresponding to the data. So if we say that in here in this image, we have a cell that is dividing and we would like to follow this event. So we draw a line always at the same place in these images. And this cell is sort of held into place in one way or another. So it's sort of attached, so forced into this shape. And so if we have a line in each of these images, we can then follow and only get the intensity along this line. So you can see that if I draw a line here, I'm going to be in the background, and suddenly I get some green, and here I get likely a bit of green and also some orange, and then I go into the green, and then I go into the background. And then I put all these lines across time. So this is the time dimension. I put all these lines into one image and create a kymograph. So you can see that the cell is not dividing. So we are in this situation. And then suddenly the cell comes out and lifts up. And so we have like, it sort of shows in a quite different way very, very quickly. But we still have one cell. We can clearly see it. And then suddenly the cell divides. And what you can see in here is that we now have two nuclei, which are in red, but we still have a bridge in between. So this is what these people wanted to show, that there was a bridge in between for quite a long time. And so they could display this with a single image, even though this is a time lapse. And then afterwards, the cells divide completely. And so we have now two cells instead of one. So kymographs are a good way to represent time as well. And if you want to publish a video, you have to also go back to the journal where you want to publish and check which codec they recommend and also which aspect ratio they recommend. So just read, it's best to, before you make your figures, you should go to the journals you're targeting and having a look. Data management is a very important aspect of publishing images. And nowadays, very often when you write a grant proposal, they ask you, what do you have for data management? How do you manage your data? And the emphasis is strongly on that. And that is because there's no point in publishing something if 6 months later, you have no idea what you were talking about. So data management and tracing what you have done to this to get this image and everything is really, really important. It's basically reproducibility of science, right, over time. So if it's time to publish and you go like, oh, where's that great image that I took 6 months ago and I presented it in my group meeting and I don't remember where it is, or sometimes 6 months, sometimes 2 or 3 years, right? And which antibody concentration did I use to get this image? Or what was the pre-processing that I applied to the image? What was my analysis pipeline? And I have the analyzed image where is the original, etc., right? So all these type of things, you must keep track of them. And clearly when you acquire an image and process an image, you don't know which one you're going to publish. And therefore, you need to do this for all the images. You need to have a proper data management system. So you need to document any modification that you have done to the image, because it will have to go to the material method sometimes many months or several years afterwards. One way of doing this, if you're working with Fiji, is to always turn on the macro recording that will systematically record the history when you build the pipeline, if you work with Fiji. But otherwise, you totally need to document everything you do. So the macro recorder in Fiji is found in the plugins, macro and record. So every time you do something, an analysis to your Fiji, it will add a line. You can save that. And save this file then in the same folder, or if you write on whatever you decide to do, but even if you make like a little notes on a Word file or something, make sure you save them in the same folder as the original image. And if you modify the image, you can put like original and name modified, something like this, so that you find many years later, you can find what you have done and how you acquired the image. One of the bad habits that people have very often is to put lots of things in the name of the file, and sometimes it creates problems, because if the names become too long, Windows, for example, but I think it's not only Windows, they cannot deal with names that are very, very long. So you don't see it until you try to transfer the images. So we try to transfer the images, then it will complain because the name is too long. So try not to put too much in the name. Omero is a great way to keep track of which image you used and how you did, and share it and annotate it and all this type of things you have seen. You can even star the images, and starring the images is a really good way for you to find many months later, oh, which was the best one of this batch? Okay, I've starred it. Okay. And we have seen now Omero Figure. It's really good. It's free, and you can use the demo server without having your own server. So you can use it. And it's also better than Illustrator, and it's so quick to work with it. Photoshop is not a very good software for scientists. It is designed for photographers. It's designed for being very creative. So it's great for that, but not for scientists. It's very easy to make serious mistakes with Photoshop without knowing it. And this may haunt you. So that is not very good, okay? So beware of Photoshop, really. I would not recommend that you use it. So this is now, I want to mention resolution. So we spoke a lot about different resolutions. And oh my God, there is yet another type of resolution. So sorry about that, but this is the way it is. We have the optical resolution, the digital resolution. We have the resolution that our image, our experiment requires. And now we're talking about printing resolutions. That's the way it is. We are in a digital world. So RGB means green, red, and blue. And so quite often you can see that images are RGB. And if we have, this is an 8-bit image, so it has 256 levels of gray from 0 to 255. And if an 8-bit RGB image will have 24 bits, and that means 8 per color, so 3 times 8. So when we print an image on paper, this is the way it looks like. If you zoom into the image enough, if you can zoom in, this is what you will see. So the printing sort of process prints dots. And so it's organized like this in lines. So this type of gradient, for example, would be represented in a printed image by a gradient of size of black dots. Okay, so the large dots in the dark area and the smaller dots in the lighter area. This is the way. And of course, if we look from far away, it just looks like a nice smooth gradient because our eyes are just not very good. So because of the limitation of the printing techniques, it's not possible to print completely black or completely white, but clearly you should not have completely black and completely white if you have no saturation and no underexposure in your images. So that's one more reason why you should not have underexposure and saturation in your images if ever you still need it to be convinced. So if you have some, then this information will also be lost. But anyway, it was wrong to start with, so maybe it's better if it's lost. The printing resolution is expressed as we had a little hint of in the Omero workshop. It's expressed in dpi or ppi. These means they are basically the same. dpi is dot per inch and ppi is pixel per inch. So depending if we talk about an image or if we talk about paper, but basically this will be the same thing. So in the printing resolution is the number of dots per area unit. Okay, so density. So this is actually a density. And it says dot per inch and pixel per inch, but really what it is square. So it's an area, it's not linear. So in reality, 300 dpi means 300 by 300, so 90,000 pixels per square inch or dot per square inch. So have a look at the requirements in density of pixels or density of dots for your favorite journal. Nature recommends 300 dpi minimum. And JCB says different dpi level depending on what you're publishing. If you have something that is black and white images, you require much higher density. But if you have color images without text, then it should be 300. And if you have scale bars, arrows, whatever, it should be 600. Unfortunately, these journals do not, and Cell is the same, these journals do not really update, let's say, their recommendations. But nowadays, there is a way to, with vector images, that you may not need to use this. But anyway, we have to follow what the recommendations are. So this is what you need to do. These are not set in stone. So if you do not have, it's quite rare that they accept something that is under 300 dpi. But even if you do not have 1000 dpi, for example, the journal will probably accept it. But 300 dpi is a little bit sort of a hard limit. So this is an image that is in 72 dpi. And this is an image in 300 dpi, and you can see that because basically it's a low resolution image, right? So we see the pixels. That's exactly the same thing as when we acquire the image. So you can see this as pixel per inch or pixel per microns or dot per inch. All this is basically the same, is the density of the dots. When so different journals print, they have a format to print their page. So basically, they're going to have, for example, a certain area of the page where you have text, and then underneath they will have some, and often the text is organized in 2 columns, for example, and then underneath they're going to have a figure, and so they have a requirement in the width of the figure. So you look at the journal, you set what it is, and it will tell you for a figure that should cover 2 columns of text, this is the number of centimeters that you should use, and a figure that would cover only 1 column of text, then this is the centimeter that you would use, or the size in centimeters that you would use, or in inches, or whatever it is. But then you need to look at the requirements of the journal. And so that gives you the size of the image on paper in the final publication, and you have to stick to that. So an image, for example, that has been acquired at 512 by 512 pixels at 300 dpi, so pixel per inch or dot per inch, it physically measures 512 divided by 300. So this is 1.7 inches. And clearly it's 1.7 times 1.7. So 1.7 in X and 1.7 in Y. And that means because 1 inch is 2.54 centimeter. So 1.7 inch is 4.3, 4.3 centimeters. So you can imagine 4.3, it's quite a small picture. This is what it will look like when it's published on the paper. You can measure it, it will look like this. So if your image, the same image needs to be published in 600 dpi, the pixel will be, or the dots will be smaller, and therefore this image is now 2.17 centimeter. That is very small. So that is too small. You cannot see anything there. So there's no point in publishing this image in 600 dpi because nobody will see anything. So if you want to acquire an image for publication, it can be a good idea to, like, where you say, okay, now I've acquired my images for my data acquisition. Now I want to have an image that actually, where the viewer can see this and that. Then I'm going to acquire with more than 512, because this way you can publish in 300 or at 600 dpi and not have to go back to the microscope to image. So as you know, we can go into Fiji, and if we look at show info or control I, we can see the number of pixels that we have in the image. We can also see it at the top. I put my square in the wrong place, it's here. We can also see it at the top of the image, the number of pixels in the image. Good, I'm trying to see how I'm doing with time. I still have 10 minutes. So the quality of the image is very important. So it's linked clearly to the compression. It's also linked to the number of pixels that you have in the area that you have, and it also linked to the software that you're using. So please do not make your figures, do not build your figures in PowerPoint. You should use Adobe Illustrator, Photoshop. But again, there's risk in Photoshop that you can modify the images without even knowing it. Inkscape is also a very good software to do this building figures, and it's free. And clearly, Omega Figure is super good, and it's easy to use and everything. So again, resizing or rotating images in PowerPoint modifies the image. And also the PowerPoint format. PowerPoint is basically based to make presentations that will be projected on the screen. So, and nobody wants to have a lag when you project. So it compresses all the images. So do not use PowerPoint to make your figures. Only use PowerPoint for presentations on screens, not for to prepare figures for printing. And when you create a a PDF, make sure that you use the best quality of the PDF. There are different types. So in Adobe Illustrator, for example, with saving PDF, you can click on options and choose the high quality printing. You should definitely use this when you want to print a poster or a figure for a journal. So now in the printing world, there are two types of images. One of them is bitmap images, and this is the type of images that we get in microscopy. And that means that if you zoom in to this image, you're going to start seeing the pixels. So this is the type we get with our microscope. And then the other type of image, so they are made of finite units, what are the pixels. The other type is vector images, which are calculated instead of represented by little squares. So in vector images, we can scale up and down, zoom in, zoom out, without seeing the pixelation. And this is very important for text, so that when you zoom in and out, it stays very sharp like this. So things are recalculated, and you remember, there's, for example, Google Earth or something like this. You zoom in, you zoom out, and then it just, the images, you never see the pixels, basically. So things like text, graphs, and scale bars and stuff like this should be in vector images. You can combine the two types of images, for example, a scale bar on top of a microscopy image. Now there are two ways. Either you keep the layers separately, so you have the microscopy image, and on top of that you have the vector image, or you flatten these and you merge them. It's called also embedded, it's called burnt. All these things mean the same thing. It means that the two layers are stuck to each other, and now if you zoom in or not or out, they zoom in and out separately. And therefore, when they are burnt or embedded or flattened, if you zoom into a scale bar, we're going to see this. They all become raster images. So a microscopy image, which is the raster type, will only be accepted by publisher if the vector layers are saved separately, okay? Do not burn, do not flatten your images before you send them to the publisher. They will not want that. So graphs and drawings and everything must be submitted as vector images as well, because there you need a very high definition since it's black lines on the white background. So the flattening is done when you save and depends on the format chosen. So it's important that you choose a format that preserves the two types of layers. So for example, we spoke about it this morning in Homero Figure, you can export as PDF files, and that means that you will have, it's a raster image. So if you zoom into the figures that you have created today, you will not see the pixels in the scale bars. Okay. So do not save as TIFF in Homero Figure because that flattens everything. Use PDF as well. Right. So you know that you can put a scale bar in Fiji, and you put some annotations, and then afterwards you can also use the BioVoxel Figure tool, which is a plugin that you could also download if you don't have it. And that is also very useful to create that. So you can use Homero Figure, or you can create this BioVoxel Figure tool to save the microscope image as a raster image with the annotation as a vector image. So VSG, it's a vector. And then you can use Inkscape to build your figure from these images. So I put here the link, and there's also a little YouTube video so that you can see how this plugin works. So now when we zoom into the image, we can see the image rasterized as it was. So all this is one of the acquisition pixels. So that has been preserved as it was from the microscope, but this scale bar is now a vector image. So if I zoom in, I can not see any pixels. So resizing an image, most of the time, since you're stuck to the width of the journal that they tell you how much it is, you probably will have to resize. And clearly in Homero Figure, it's easy to do, but do not resize several times. If you resize once and you save, and then you go back to this image and resize, no, go back to the original. And clearly it's an advantage to work in Homero Figure because you always work on the original anyway. And otherwise, they are in Fiji some ways. If you go to image adjust and size, you get this guy here, which shows you the number of pixels. You can change the number of pixels. For example, go from 256 to 128. So in this case, the image size changes. So it's okay to change the number of pixels to a lower number. The image quality will look down because it's a little bit like binning. You make bigger pixels, basically, but not good to go higher because then you're creating data. Your image will look smoother, but this is actually artificial. So the things that you can change in an image is the number of pixels. You can change the physical size in inches or in centimeters, or the DPI resolution. These three things depend on each other. So in here, we are changing the number of pixels in the image. We're using this technique, but we keep the same DPI. And so the image goes from 512 pixels to 256, and because therefore we have a smaller image. So yeah. So they are calculated from each other, so you can only change two of them. But if it's acceptable to go down in the number of pixels, but not to go up. So do not increase the number of pixels you're creating data, whereas going down, you lose quality, but it's anyway acceptable if you need it in the image. So the only way to get a larger image with the same DPI is to go back to the microscope and re-image with a higher pixel density. So this is the type of things you need to think about when you acquire the images. You can change the physical size of the image and the DPI in Fiji using image adjust scaled to DPI. And this is what it looks like. And so you have here the size in inches and here the DPI. So you can change. So this is the image. You can see 512 by 512, and I stay with the same number of pixels. But now when I change the DPI, the physical size of the image is changed. Okay, so I go from 600 to 300, and so the image is going to be double the size, but at 300 DPI. So this way you can change the size of the image. When you downsize the number of pixels, we have the same issue as for microscopy. We go from a high resolution to a low resolution image, and therefore we have the same issue exactly where we can create aliasing artifacts that you can see in here. For all the details that are not resolved anymore, you can get aliasing artifacts. In this case, because we have a clear structure in the sample, then we see moire effect, so it's easy to notice it. But even if you do not have any clear structure, then you're going to create aliasing artifacts anyway. Okay, so always keep track of all the processing steps that you have done for each image. And again, you don't know which image you will use for later. So you must think of how you organize yourself to be able to find which processing steps you have done several years later, okay? So that when you publish that image, you know exactly what has been done and you can put it in the material and method. So this requires you to be organized from the second when you actually, before you acquire the image, because you need to also be able to tell what has been done on your, for the sample. So you need to organize this before. And so Doug, who will talk later today, said something that stayed with me, and I think it's very clever. He says users can do just about anything to a digital image as long as they provide a detailed protocol of what was done to retain the unaltered data and retain the unaltered data. If the protocol is out there, they might be called an idiot based on their image processing protocol, but they cannot be called a cheat. And that is something that we do not think about too much, but I can tell you from that the longer you stay in research, the higher the risk that you are going to be questioned about something that maybe you published several years before. And I know several people, including me, who were in that situation. It is not fun. And even if if you have processed something and done it wrong, it's one thing as long as you have the original data and you can justify what you have done, even if you made a mistake, it's okay. But if you have not documented, this means that you may be told that you did it on purpose. And that is really tough because when you didn't know what you were doing, basically, it's very tough. It's also very difficult to remember the way you were thinking several years before. So you do not want your paper to be scrutinized by someone who questions the integrity of your data, often many years after they have been acquired or published. So this is 100% not fun, and it will take a lot of your energy. So make sure that you document everything that you do not have things like like saturation, underexposure, bleed through, all these type of things. You must avoid them completely and write in your papers that you set your settings in such a way that you avoid all these macroscopy artifacts. And you do not want your figure to be rejected because the images that you acquired 1 year earlier are heavily underexposed. And journals, of course, have ways to, and they screen the images that you're sending so that they do not want to publish anything that is wrong. So they have a certain number of screens. And even if you do not manipulate your images on purpose, if people discover this, you will get in trouble. So please be careful. That is it for me now. So I would like to know if there are some questions about all these details about publishing images. Yes, Aphrodite.
Sylvie, may I ask? I noticed that when I finish with adding the scale, and then I flatten the image. Then I save as TIFF, usually to use it on a PowerPoint. Notice that there is a blue line on the edge of the image.
Yes.
I could share my screen to show it clearly.
You do all this in which software?
In ImageJ. So here you can clearly see this blue line, and it's created in all the images.
All your images, yeah.
Why
So,
this happening?
yeah, I'm not sure. These are acquired in NIS-Elements, right?
Yes.
Yeah. So somehow there must be like 1 or 2 pixel. The image is like a couple of pixels bigger than the stuff that is acquired. Which system did you use to acquire these images?
It's a spinning disk.
It's Victor.
Victor. Mm-hmm.
I think it's good
this
that you
is a large
record.
image, right?
I'm sorry?
Is it a large image?
Yes, it's an overlay.
That's why.
Yeah.
That is why, yeah, because you know when you take a tiled image, JJ is totally right. So when you take a tiled image and then you stitch the image together, the tiles will be slightly rearranged so that the borders match. And so therefore, there will be some areas where the big image will have no pixels, and that's why it's black. And basically, when you, I think that in your image, you might still have the high-low, the mask that shows you the saturation and underexposure.
And that's why it's blue, because...
That's why it's blue, because it's basically there's no data at that place.
Yes. Okay.
And so the intensity is 0 there, and it shows in blue. So I think that if you remove the high-low color mask, maybe this will disappear.
Okay. And something else, how can I keep the image with the scale to be vector and the image to not be, of course, not vector? When I don't save it as TIFF, I see a black image because of the layers.
Yes, no, it's not because of the layers. It's because the image is in 12 bits. So, basically software like PowerPoint or anything, they cannot read the 12-bit image, or they can display it properly, the 12-bit images. So what you need to do is save it in 8 bits. On the if what you want to do is put these images in a PowerPoint presentation, there is a very good function in NIS-Element. If you just press the X key, so you display the image the way you want. Maybe you want, you know, each color. So you split the image in the different channels. Then you have an overlay, and then you have the channels in black and white, and you put your scale bar, whatever. You display it the way you want exactly to show, and then afterwards you press the X on the keyboard, and that will create an RGB 8-bit image.
Okay.
That you can save in any format and just insert it in your PowerPoint.
Okay, great. Thank you.
You're welcome. I didn't think about that thing, JJ, so that was a good thought. Are there any more questions about publishing images? So we're going to go, if there are no more questions, we're going to go into the other Zoom and have a discussion there. I see you guys there.